Welcome to the gene annotation tutorial. I'll go over step by step how to complete this assignment. First, you can find the assignment under your TA's Canvas page under Assignments. By clicking on this, it'll direct you to download two documents, one called Bacterial Gene Annotation Exercise, which is the document that your TA went over in discussion, and another called Bacterial Genes for Genome Annotation. And this is just the list of genes that we will be annotating. So you are each assigned a specific gene according to your last name. For example, my last name starts with the last letter Z, so I'm assigned the last gene. So I don't give away any answers to you T through Z people. I'm going to actually do this tutorial on a different gene. This monstrosity of a gene. So the first thing you want to do is you want to copy your gene and paste it into a new document. And then you want to label the header with your name and your section number. Now for this assignment, you'll need a few things. You will need the name of your gene, the annotated DNA, the annotated mRNA, the picture of the hairpin terminator, a description of whether your terminator is row dependent or row independent, and finally the protein sequence. So let's begin. The first thing we're going to do is we're going to name the gene. Every year students lose points because they forget to do this simple step. So that's why we're going to start with this. To name the gene, we want specifically the protein it encodes and what organism it's from. To do this, you're going to have to do two BLAST searches. So if you remember from your BLAST assignment, you'll go to NCBI to access this tool. So let's copy our gene. And then we'll go to NCBI. NCBI BLAST. Now, the first BLAST we're going to do is the BLAST X because we want to know what protein this gene encodes for. So go ahead and paste your gene and then leave everything as the default setting and then hit BLAST. Now, this BLAST usually takes a while because it has to translate your gene and then match it with whatever protein. So don't refresh, just be patient. I already had one cooking, so I can bring it up right here. And it appears that our protein is protein disaggregation chaperone, and from the species E. coli. So let's take this protein disaggregation chaperone, copy that, and then we'll put it in our document. So the first part, the protein, we got it covered. So this particular BLAST did give us the species of our organism, but we do want a little bit more specific information, so we're going to do another BLAST search. For other BLASTs, sometimes it gives you a non-specific species, like multi-species. This is especially when you want to do the next BLAST, which is a nucleotide BLAST, in order to pinpoint which species it's actually from. So we'll go back and then let's just run NCBI BLAST, nucleotide BLAST, and then I need to recopy the protein or the DNA sequence. Whoops. Oh my God. There we go. And then you could leave everything as a default and then hit BLAST. So this generally doesn't take very long. Now, it definitely is E. coli. We don't want this synthetic E. coli because that's just a man-made protein or a man-made genome. So we're going to take this E. coli MG 1655. So we'll do this and copy this. We don't need the complete genome. It obviously matches with the complete genome the closest. So that's not important to us. we can include this. And there we have it. The name of the gene is complete. Now, the next thing we want to do is annotate our prokaryotic DNA. For this assignment, we have to find a few sequences. We must label the -35 and -10 regions of the promoter, the Shine-Delgarno sequence, the coding region, the terminator hairpin, and the AT region after the hairpin if your terminator is row independent. The assignment provides you a color key on how to annotate each section. So let's look at this color key. It should be down here somewhere, right here. So go ahead and copy and paste this into our document. The reason you want to do this is because you want to follow these guidelines very closely and make sure you're using the exact same colors and formatting. This will make things much easier for your TA to grade, and happy TAs are way nicer than angry TAs. So the directions tell us that the DNA given is the coding strand. We know that the beginning of the strand is then the 5', and the end of the strand is the 3'. So this gives us a good idea of the orientation, knowing that our promoter regions are going to come before the coding region, and then our terminator regions are going to come after the coding region, so somewhere at the end. So then that means the first thing we want to do is actually find where our coding region is. So in order to do this, we're going to use XSpaCy's translate tool to translate our DNA sequence. So we're going to go look for the link to XSpaCy. Aha, here it is. So go ahead and copy this, and we're going to paste it. Get rid of this stuff. Let's paste it here. All right, so we're going to need to enter our DNA sequence. Let's go back, copy all this. Copy. All right. So once we paste in our DNA sequence, you want to make sure your output format is verbose, and then you can hit Translate Sequence. So the output, or the results that it gives us, is 6 different reading frames, 3 in the 5' to 3' direction, and 3 in the 3' to 5' direction. So they're pretty much inverses of each other. Now, the reason it gives us 3 different reading frames is because you know a codon to be a set of 3 nucleotides. So shifting the reading frame by 1 nucleotide gives you a new reading frame with new sequences of amino acids. By the 3rd shift, you pretty much reestablish the same reading frame from before you shifted, so it just goes back to square one. Now, when we're looking for the coding region, we want to find a region of the DNA that may code for a reasonably sized protein. The median length for a prokaryotic protein is 267 amino acids long. So we want to look for the longest region of translation where a MET starts translation followed by a string of amino acids and a stop. For this gene, you could see right away that the longest region is in frame 1. I don't see any longer regions than this. So since the assignment wants you to include the protein sequence, you can copy the sequence from here and paste it to your assignment. So looks like MET starts here, stop ends right here. So we can copy this, bring this back to our assignment, down here where we have the protein sequence. And since I'm like slightly OCD, I'm going to remove the formatting. All right, great. So now we want to find where this translates to on the actual DNA. So to do this, we're going to go back, we're going to hit the back arrow, and then we're going to change the output format to includes nucleotide sequence. Then hit translate, and this view will give us the amino acids and the nucleotides. The only problem is that it gets rid of the pretty highlighted region where it shows us directly where our coding region is. So if you've forgotten, you can just hit the frames, and I remember the longest frame to be in frame 1, so I'm going to hit this, and it'll show you it again. So now I can look closely and see that our protein starts with MGGV. So go back, MGGV right here. So I'm going to take it starting from the ATG, which I know to be my start codon, and then I'll just extend it a little. I'm going to copy it, and now I'm going to go back. I'm going to search for it in my document. Aha, here it is. So this is the beginning of my coding region. So based on this key, I'm going to make it red, bold, and underline. Great. So now let's go find the rest of the region. So I'm going to go back to this. I'm looking for a VAVQ. So I'll go back. It's probably going to be down here somewhere. VAVQ. Perfect. So I'm going to take it up to the stop codon. Include your stop codon in your coding region. Remember to do this. And I know that a stop codon is UAA. So I'm going to take it up to the last A. Go back a little bit. Copy this. Now I'm going to look for this in the document. Aha, here it is. So this is our last region. So I'm going to take everything in between here and bold it red and underline. So just make sure I get everything. It's a rather large coding region. Okay. Almost there. Okay. All right. And there we have it, our coding region. Now, if it was difficult for you to visualize the longest sequence using the Translate tool, you can use this other tool called ORF Find. This program is nice because it lets you reduce or increase the amino acid reading frame, so you can sort of look at some different options. So let me just show you how to use this real quick. It was under this right here. So we'll copy that, open another window. Okay, so now we have to copy our DNA sequence again. We'll be doing this a lot. Okay. All right, and then you can hit ORF Find. So this gives you a nice graphical representation. You can see that this is the longest protein. You can also change the reading frame. So now it's only going to show you the ones with amino that are at least 300 amino acids long. Now it's going to show you the ones that are at least 50 amino acids long. So this kind of reduces some of the options that you have to choose from. And so I'm going to go ahead and choose our longest. And again, it gives you both the amino acid sequence and the nucleotide sequence. So you can see it matches pretty much what we found. So that's all well and good. So know that you have that tool as well. Now we want to annotate the rest of our DNA. So next we're going to find the Shine-Dalgarno sequence. The Shine-Dalgarno sequence we know from class to be represented by AGG. Now, the important thing you must know about the Shine-Dalgarno sequence is that it's actually a consensus sequence. A consensus sequence is a short stretch of nucleotides that possess considerable similarity from one organism to organism. This suggests that the sequence is specific to an important cellular function that is common in all organisms, and in this case, the initiation of translation is what the Shine-Dalgarno does. So that's very common in all prokaryotes. This means that you're not going to always see the Shine-Dalgarno as AGG, AGG exactly. Often 1 to 3 nucleotides may be different. So let me just show you an example of this. So here I have 4 different organisms. The first one has regular AGG, AGG, Shine-Dalgarno. The second one, the Shine-Dalgarno, is AGG, AAA. So you can see 2 nucleotides are different. The third one is CAG, AGG. Again, 2 nucleotides are different. The fourth one is AGG, AGU. So here the last nucleotide is different. So there is some variation, and that'll help you find your Shine-Dalgarno sequence. So let's go back to our sequence. So since it's necessary for the beginning of transcription, it'll be before the start codon, and it should only be around 2 to 11 nucleotides away from that start codon, so around this region. Remember, because the Shine-Dalgarno is a consensus sequence, it's not a set number of nucleotides away. So let's just look upstream and see if we can find anything that looks like an AGG, AGG. I actually don't see anything. So if this is the case for you, it's likely that your AGG, AGG is somewhere in your coding region. So assuming this, that means that this first start codon is not actually my start codon. So I'm just going to look downstream. I don't want to shift the reading frame if I don't have to. So I'm just going to count 1, 2, 3, 2, 3, 2, 3, 1, 2, 3, 1, 2, 3. All right. So here's another ATG. This could be my actual start codon. So let me just get rid of this and make this back to black so it'll help me see it better. Okay, so if this is my actual start codon, now let's see if I can find a Shine-Dalgarno sequence. So looking down, I see a GGG, AGG. Oh, but I also see an AGT here. Okay, and then before and after, there's only this 1 nucleotide difference for each. So this is kind of a crapshoot. I think for this purpose, I'm going to actually go with the GGG, AGG as my Shine-Dalgarno because it gives me a little bit more of a gap between my start codon. So I'm going to pick this one. So pink, right? Pink and bold. Pink, bold, underline. All right, perfect. So I went with this one. So since I had to shift my start codon, make sure you make that change in your protein sequence. So this met is probably going to be my first start codon. So I'm going to get rid of this, and now we're good to go. Now I want to say something very important about finding your Shine-Dalgarno. If you see that your Shine-Dalgarno is right next to your ATG, it's probably your first ATG is probably not going to be your starting codon. Your starting codon is probably going to be somewhere else downstream because there needs to be a gap between your Shine-Dalgarno and the starting codon. So just keep that in mind. Don't be afraid to shift your starting ATG if you have to. All right, so now that we found the Shine-Dalgarno, the next thing we're going to do is we're going to find the -35 and -10 consensus regions. So we know that the sequence for -10 to be TAT-AAT and for -35 to be TTGACA. Now, keep in mind that these regions, like the Shine-Dalgarno sequence, are also consensus sequences. So they will not always be exactly what you expect. They may also differ in 1 to 3 nucleotides. So first, I want you to look upstream and see if you see 6 nucleotides that kind of resemble the first -10 sequence, the TATAAT. So we're just going to go up, take a look. Let's see. Nope. Ooh, here's something. TTTAAT. Now that sounds pretty close. That only substitutes this one T for an A, or A for a T. So I feel pretty good about this. So I'm going to blue underline and bold this as my -10 or -10 region. So next we're going to look for our -35 region, which has the consensus sequence of TTGACA. Alright, so we're going to look even further upstream. So oh, here's the TT. GAGG. That's pretty close. TTGACA. So like only the last two are different from our consensus sequences. That's fine with me. So I'm going to green this, bold, and underline. Now, the -10 and the -35 are generally 10 to 18 bases apart. So ours are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 bases apart. So that's pretty good. We're on the right track. If you're having a bit of trouble finding the promoter regions, luckily we can use a program called Beprom by Softberry, which is found in your document. So we'll go down, right here. So let's try using this sequence to find our promoters. So open a new window. Alright, so I need my nucleotide sequence. Now, something that might be really helpful for this is if you only take the sequence before your coding region, because this is where we expect our promoter regions to be. We don't really want any other promoter regions inside our coding region because that's probably not for this gene. It might be for another gene. So we don't really want to confuse ourselves. We just want to filter that. So we can do that by just taking this initial sequence. So then we'll go here, paste it, and then hit process. All right. So we have two results. We have a -10 and a -35. Our -10 is at location 235 and it's TTGAATAAT. And our -35 is at 211 and it's TTCACA. Now notice that these are different than the ones that we found when we were looking upstream. And that's probably a bit confusing. Which one should we use? Well, remember that Beprom is only 80% accurate. So if you found a pretty solid promoter region, you should probably just trust your eyes. However, know that we will accept either answer. So if you decide to go with this answer, we'll accept it. If you decide to go with the answer that you found by just scanning, we'll accept that. If you really can't find it by scanning with your eyes, I would go with the Beprom answer for some sequences because it's pretty hard to tell. Here, I feel pretty confident about the answers that we found, so I'm just going to stick with these and move on. But it's really up to you. Again, we accept both. So, next on the list is the terminator. We know that there are to be two types of prokaryotic terminators, row-independent and row-dependent. Both use hairpins for termination. The difference between the two is that row-dependent terminators use a rut sequence, which is rich in Cs, and row-independent terminators use a string of uracils that are immediately following the hairpin and destabilize the DNA and RNA pairing to terminate transcription. For the purpose of this assignment, we will not be looking for the rut sequence. However, we can use a program called Arnold to see if there is a palindromic sequence followed by a string of uracils, indicating the presence of a row-independent terminator. So, we can find the Arnold program down here, right here. So let's copy and paste this. Okay, so we're gonna need our DNA sequence, surprise, surprise. So let's take the whole thing. Remember to take the whole thing, because otherwise it might give you the wrong answer. So, paste that. Now, be sure to select the options forward, yes, and change. Now we can go ahead and run it. All right, so it does give us a predicted transcription terminator. The blue regions are the palindromic sequences, and they form the hairpin. And then you can see that immediately following the hairpin, there's a string of T's. This red sequence isn't part of our hairpin, it's actually the loop part, so we don't want to underline that. So let's copy this. Let's find where this exists. All right, here it is. So since this is part of our hairpin, we want to double underline this. Perfect. Now let's go back. Let's find the other part of the hairpin. Ooh. Okay. Nope. Okay. All right, and here it is. So I'm going to double underline this as well. Now, since this is a row-independent terminator, that means the hairpin is followed by a string of T's, or U's in mRNA. And here it is. So we want to double or single underline this. All right. So this is all the annotation you need for this type of terminator. Now, let's pretend that the Arnold program said the total number of predicted terminators is 0. So this means that we likely have a row-dependent terminator. And so we're going to need to use another program called QuickFold to find out where our terminator is, or our hairpin is. Now, if you have a row-independent terminator, like we do right here, then I still want you to do this next step, because the picture that you're going to get has to be included in your assignment to receive points. But know that the actual underlining part in your annotation should be based off of what your Arnold result was, not what your QuickFold outputs are. So let's go ahead and bring up QuickFold. It's up here, right here, QuickFold. All right, perfect. So let's grab our DNA sequence, like always. Now for QuickFold, since it's looking for the possible foldings of your mRNA, you only want the end after the coding region, because this is likely where your terminator is going to be. If you put in the whole region, you're going to get some crazy picture where you're not going to be able to see anything, because it's going to try to determine the stability of their whole DNA folding. But we only want, that's not really helpful for us at all. So we only want the end of the sequence for a clear picture and the right picture. So we'll take this, we'll paste it, And then the only thing you want to do is change your energy rules from DNA to RNA 2.3. And then you're going to hit submit. Alright, so the results gives you a list of possible foldings with different delta G's. You want to pick the one with the most negative delta G because that indicates that it's most stable. So it's generally going to be the first one. So go ahead and open up this image. Perfect. Alright, so this is a picture of possible hairpin structures that you're going to get at the end. Now, remember that our particular sequence is a row independent terminator. We already know that. So we don't want to try and find where the hairpin is. It's probably not going to be right, actually. Yeah, because our hairpin, I think, was like right here or something like that. So, but we do want this picture. So we're going to copy the image. And we're going to paste it right here. Awesome. Now, let's pretend that it was a row-dependent terminator, that we didn't get an Arnold output. So then, what I would do is I would try to find the most prominent hairpin structure. So I already see this. This looks pretty prominent. And I would try to find it with the most GC pairing. So this looks like it actually has a lot of GC pairings. So we're on the right track with this. So whenever you want to actually annotate your DNA, you only want to take the regions of the hairpin. So it looks like this region, this region, this region, and this region. The easiest way to do this is literally search TATAAT in your DNA and then just double underline those regions. Now remember, because there's generally not a string of T's after a row dependent terminator, you don't need to double underline, or you don't need to single underline the row of T's. So you guys are probably going to have a lot of questions with this step because generally you're going to get a crazy picture with hairpins on hairpins and you're not going to know which one to choose. Now, to mitigate this, what you could do is you could go back to your list and try to find a better image. So you can choose one with a lower delta G and see if you can find, a prettier image where you can actually see something. Now, know that we understand that it's very, very difficult to actually find a good hairpin. It might not be even feasible with some of your pictures. Just try your best. However, we know your struggles with this part, and we've done it too, so we definitely struggle through this as well. Thus, we're generally pretty lenient with our grading on this part. So, really just try your best. Now, a few things about this program to put you at ease. I just want to mention that the program is extremely variable in the foldings that it gives for the same sequence, due to the fact that the DNA is very flexible. So you could go in one day and get a completely different picture than if you did the same sequence submission the next day. To prove this, I actually went in on 3 different days and I got completely different pictures, and I put the exact same sequence in. So this just goes to show that you really shouldn't freak out about this. Us TAs, we don't even have the correct answer or the right answer. So the best we could all do is just go with the rules, pick the most prominent hairpin with the most GC pairings and call it a day. So, for example, for this one, I would pick this hairpin. And then this region obviously would be the 5 or the 3' UTR, right? And then I would pick this one for this particular picture. And I would pick this one for this particular picture. But like I said, you know, don't freak out. Just pick the best one. Just give it your best shot. That's all we can ask for. So, hopefully that puts you a little bit at ease for this step. We usually get the most questions on this. Now, don't forget to quickly describe whether yours is a row dependent or a row independent terminator. So, for example, for us, we have a row independent terminator, likely because our hairpin is directly followed by a string of T's or U's, depending on if we're talking about our mRNA, that destabilize. Oh, I can spell really well. Destabilize the DNA and RNA pairing, thus ending transcription. Cool. That's all you really need to say. And if you have a row-dependent terminator, you can say that there isn't a string of T's followed by your hairpin, or after your hairpin. So that's really all you need to say. Now, the last and final part of this assignment is to include your annotated mRNA. The easiest way to do this is just to copy the sections that are part of your transcribed mRNA from your DNA and replace the T's with U's. So remember that transcription occurs after your promoter regions. We don't really have a set number of nucleotides between the -10 region and the +1 region where transcription starts. There really isn't a good answer for that. But for the purposes of this assignment, you can just assume that transcription is going to start 10 nucleotides from the end of your -10. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let's just say that transcription starts there. Now, transcription will end. Transcription will end at the end of the string of T's for a row independent terminator, and at the end of the hairpin for a row dependent terminator. So since we have a row independent terminator, we're going to stop it at the string of T's. So copy. Now we can easily just replace it by starting a new document and then edit, find, replace, search for T's, replace with U's, replace all. And there you go. Now our DNA is mRNA. So let's just put this back and enter it right here. All right. So looks like we've got everything. We've got the name of the gene, we've got the annotated DNA, the annotated mRNA, the description of whether it's row dependent or row independent, the picture of the hairpin, and the protein sequence. So that's it. Hopefully I was able to answer all of your questions. I'm very, very sorry if I was not. Hopefully you enjoyed watching this whole process and stuck with me throughout the video. If you have any questions, please direct them to your TAs. Otherwise, thanks for watching and good luck!